this. DNA is made of two strands. That's the point of the styrofoam, and there's lead in it. Lead, styrofoam, little doohickeys here. One strand, all this DNA, this is DNA. Let me use a different color. From one side to the other is DNA. That's one molecule, technically a single molecule. One single strand of DNA. And it's a molecule because they're covalently bonded. They're all sharing electrons. Very strong bonds, all of them. They're not going to be that strong in the models that you're building because there's little magnets that are holding them together. Then the other strand is going to be, it's going to be matching, and it's going to twist around. And that's why you have this. That's why they give you the two styrofoam things to play around with how they twist around an axis. So this is a second strand. Second strand of DNA. Together, biologists call this a single molecule. One molecule of DNA. Technically, it's two molecules, but it gets confusing to people, right? Why do we say it's two molecules? Because this one is covalently bonded to each other, and this one is covalently bonded to itself. Is that making sense? That becomes important later when we do some DNA technology, but what's holding the two strands together are these rungs, these nitrogenous bases. And I'm just going to do them all in green. It's about, this is obviously not accurate, at least I hope it's not, you understand it's not accurate. This DNA molecule is on your logo, your school logo. This green is called a nitrogenous base. And I told you that to have a language, to have a message, you have to have variances. We all agreed on that, right? You have to have differences. This is your differences. This is where this is where you find differences. You're going to have one of four options here. There's one of four options. This is very important. This is where you find the differences, these nitrogenous bases. You have four options. You have A, hydrogen bonds with T. Let me not put lines, because lines mean covalent bonds, right? So let's not put lines. Let's put dots. These are hydrogen bonds. I'll talk about what that is in a second. A, hydrogen bonds with T, and G, hydrogen bonds with C. How many hydrogen bonds are there between G and C? Three. I see you're reading the dots, which is fair. But the only reason I'm putting dots is so that you don't think it's a covalent bond. All right. These aren't three covalent bonds. These are just attractions. Remember we talked about salt, ionic attractions, right? We said sodium and chloride, one's positive, one's negative. They stick to each other, but they're not sharing electrons, correct? This is more weakly, it's weaker than this attraction, but these two are weakly attracting each other, like weak magnets. These are like strong magnets. Does that make sense? Okay. But this is how you're going to have to pair them. Now, there's a problem with these kits. I got them this way. I didn't make them. But they were mislabeled. So some A's have been labeled T's, some T's have been labeled A's, some C's have been labeled G's, etc. Yeah.
Can you zoom in on the part where it says DNA?
Here. Here?
Yeah.
So in order to solve that, you're going to have to use these facts. It actually turns out to be serendipitous. Do you know what this word serendipitous means? Say what? Covalently bonded. Each of these strands is covalently bonded to itself. But look, I can separate them. How come when I pull these apart, the strands don't fall apart? Because each strand is covalently bonded to itself. Does that make sense? But they are hydrogen bonded to each other, so they do kind of stick together. But because that sticking together is weaker, I can separate them. Does that make sense? That becomes very important. We'll see why in a little while. Is that good? Can I go on?
Can you go over?
In a second. Can I go on? Okay. So A and T match, G and C match, you're going to have to figure some of that out as you're building your kits. And today you're just supposed to have some fun with it. But A and T, here's two tricks that are going to help you figure this out. First of all, G and C are going to have 3 hydrogen bonds. And the second clue that's going to help you solve this mystery is that always in DNA, a double ring nitrogenous base pairs with a single ring. Always. So you never have 2 single rings. You'll see this. You may not understand what I'm saying right now, but when you see the kit and you actually see the double rings, you'll understand. So if you have 2 single rings trying to stick to each other, they might stick to each other because they both have 2 hydrogen bonds, but it won't work. Your DNA molecule will look weird because it won't. And energetically, in the real world, that wouldn't work at all. It would just fall apart. All right. So A always bonds with T. That's part of how your DNA always stays the same. That's how you edit your DNA. Since they always match, and if they don't match, they don't stick together. If they don't stick together, it doesn't work. That's how you make. It's one of the ways you edit. Makes you spell check your DNA. That's how you have 1 trillion cells with all the same DNA in it. Think about it. You have 1 trillion cells. Imagine copying one book. How accurate would you be if you had to copy a book by hand?
Like exact copy.
Exact copy. How accurate would you be? You'd make many mistakes, wouldn't you? And not only would you make many mistakes, can you imagine doing that 1 billion times? 1 billion times. That would be a trillion. 1 trillion, because you have 1 trillion cells. How many mistakes would you make then? A lot. But your cells can self-correct because A doesn't go with T and A doesn't go with C and G doesn't go with T. So, and there's other ways the cell corrects, but just know this. And then the other thing you need to know before I let you loose on these kits is that you have, and we already talked about this, but I want to review this. In one strand, I'm just going to use black and red because that's what I used up top. Okay, maybe I'm not going to do that. See, why do I have to quit and come back in? Why? There's no reason. Just machines hate me. Apps hate me, like the TV. Anyways, what we do is we have your deoxyribose. You've already made this, right? You made this in class. Is that correct? Some of you have to finish making it, but you have mismade. That's why I had you make it, because it's important. Deoxyribose. You have them numbered. You numbered them. I numbered them for you several times in class. Go back and review the previous postings. on this one, carbon number 1, you're going to see comes off of carbon number 1 a nitrogenous base. What does that mean? Either a single ring or a double ring, an A, T, G, or C. One of those 4 bases. Is that clear? This is your variance in the language. This is where you get your letters, right, for your words. But there's only 4 letters here. Then on this end, on the 3' end, you have, obviously you have hydroxides on all the carbons, just like you made, right? But when you're done, when you're putting them together, they connect to a phosphate. We did this, didn't we do this already? Sugar phosphate backbone, I put it on the board, right? Okay, yeah. And then connected here above is another phosphate, and you have this chain of sugar phosphate that stays the same. These are all the same. These don't change. They make a chain of nitrogenous of sugar phosphate backbone that is connected using covalent bonds. Now, when you're connecting them, and now off of these ones are all these nitrogenous bases, A, T, G, or C. And there's going to be different ones depending on your code. This is your code. This is what makes each of you different, is this difference in code, A, T, G, or C. When you're putting these 2 strands, this is going to be one strand, right? This is the other. They're going to hydrogen bond, but they have to be upside down. You're going to see that they won't work. If they're right side up, they won't work. They have to be upside down. Is that understood by everybody? And if they're upside down, then they just like in a real DNA molecule, because these are using magnets, if they're upside down, they're aligned correctly, they will snap together. 2 if it's A and T. 2 if it's A and T. And 3 if it's G and C. All right? And they're going to twist it like these two things twist. They're going to twist into this double helix, and you're going to see the double helix form if you do everything right. And it's obvious when it works. And the rungs, whenever you see these rungs, you'll see these little ladders. They call it DNA ladder, right? Because you'll see this ladder. The ladder are the rungs. Do you know what a rung is? The steps of a ladder. The steps of a ladder, rungs of a ladder, are the nitrogenous bases. One side has their nitrogenous base, the other side has theirs. They pair up and they hydrogen bond to each other. They have these weak attractions between them. It's important that you understand they're weak attractions compared to the covalent bonds between them. But what happens if you use 1,000,000 weak attractions? Do you think it's going to get strong? Yeah. 1,000,000 little stickers can hold a lot together. Because even though each sticker can only hold, 2 pounds, half a pound. You ever see those command strips? Each command strip can hold 2 pounds. What if you want to hold 20 pounds? What do? Okay, multiple command strips. Put 10 of them on it. Now you can hold 20 pounds. Does that make sense? So this holds a lot more force because there's so many little hydrogen bonds. All right, so now if you haven't finished building ribose or glucose, build those using these models. If you're going to use DNA, the DNA kits are back there. They're labeled DNA kits. They're stacked. One of them is missing the stand. You connect the stand, bring it over here, and I'll show you anything you need to know. And you're also missing these little styrofoam things because they didn't put them away from last period. All right, that's it. I'd like not to talk to you so you can give you a break from my voice, and you guys can have some fun. Remember, there's no stress today. Just you building, and then Monday we'll go over it again and take it step by step. Any mistakes you make today, don't worry about it. But think about it. Maybe take some notes and say, I wonder what I did wrong. And make a note, some observations of what you did. But have fun, though.